Coach John, who's calling in from the City University of New York. George is a professor of chemistry and biochemistry at City University of New York, and his research is highly interdisciplinary, focused on the molecular design of synthetic lipids, membrane mimics, soft matter, trans fat alternatives, and organic materials chemistry. He holds several patents on innovation related to the value-added chemicals, surfactants from renewable resources, and their potential utility in food and biomaterials. So he's going to be talking about possible future directions in terms of research.
Okay, thank you, Emily, and sorry for a couple of minutes, and thank you very much for the invitation to speak about this one. I'm supposed to be there, but unfortunately I'm still in New York, and I'm happy to talk about today's subject, the eco-friendly chemical herders. And we heard about a lot about herders, but I will talk a little more about fundamental science, and this is from City College, and this is a collaborative work between chemistry and chemical engineering. And first of all, I start with the acknowledgement for the Gulf of Mexico and for NSF for funding. And the fundamental question is why the oil is spreading on water. And I'm sure all of you know that one. It is much more of a surface tension, because the surface tension of water is higher than the total sum of the surface tension of the oil-water interface, and therefore the oil is spreading on water. And right now, because of that spreading of water, it creates oil spill. And these are the typical standard procedures for mitigating oil spills. One is burning or in situ burning, and dispersants, booming. These are the typical traditional ones, and there are a couple of them, skimming and bioremediation. And I will talk mostly about how we can generate new generation herding agents as well as skimming or gelation technology, starting from small molecules which are surfactants, which are derived from biomass or eco-friendly oil collecting agents. The first project I will briefly talk about phase selective gelation technology. In this case, how we develop these molecules or surfactants to solidify oil in presence of water. We start with byproducts and co-products or waste materials which are derived from plants and crops, and we try to do some very simple chemistry. It may be a one-step chemistry, or we do biocatalysis, much more eco-friendly. We are creating molecules, and those molecules are all of with a head group. It is typically hydrophilic or water-loving. Then we have tails attached to those hydrophilic particles, and these are our hydrocarbons, relatively small hydrocarbons, maybe 5 to 16 or 18 carbon atoms. And this is coming from vegetable oil. We are connecting them by this simple chemistry, creating simple molecules. And these are our building blocks. And these molecules, when we keep it in water, in order to minimize the energy, the surfactant molecules self-organize to create different kinds of structures and functions. And depending on the structure of this molecule will self-organize to create fluid structures like micelles and liposomes or vesicles. Sometimes we are getting jelly structures, sometimes we are getting liquid crystals, and sometimes some of these molecules have the capability to herd the oil. And I will talk about those little things. As I mentioned, all these molecules having a head, which is water-loving, and there are tail, they are oil-loving. And we call them amphiphiles, or in the shape of a tadpole, and with a head and a tail. It is typically a soap-kind molecule. Once we have the soap molecules, if you put it in water, we are getting micelles and vesicles, and sometimes they happen to be a bilayer. And here you can see a simple bilayer. It is just a bilayer. It is typically in our body. It is bilayer membranes are there. And if it is happening in the oil base, we'll get a reverse bilayer. And depending on the structure of the surfactants we are using, sometimes we get three-dimensional network, just like a sponge. A piece of sponge which is created by fibrous networks, and these fibrous networks, sponge can congeal the solvents, and we are creating jellies. And we have the capability to make or design molecules which can potentially useful for making water-based gels, just like the gelatin jelly, the dessert what we eat, or we can create oil-based gels, and we are using this technology for oil thickening. And where from we are deriving all these molecules, you can see these are all simple sugars, and this sugar is, again, this thickelose is typically used for baking of a cake, and sorbitol, mannitol, and xylitol, these are simple sugars, and the cheapest sugars, they are typically found in chewing gums. They are edible, and we are using those kind of sugars to create simple amphiphilic structures. This is the head part, this is the tail part. It is mostly coming from vegetable oil. We are creating molecules with a head and a tail, or in other words, it is surfactants. And these surfactants have the capability to solidify oil in presence of water. Usually, if you in a laboratory or at home, if you have a oil-water mixer, if you are adding soap into that one, it is typically creating kind of a white, milky product. In our case, this particular family of molecules have the capability to solidify oil in presence of water. That's why we call them phase selective. It can selectively migrate into one phase, oil phase. It can solidify. If it is happening, this is our dream. If there is an oil spill, we can spray our simple molecules in simple, typically in alcohol or some those kind of solvents, which can solidify. We can skim it off. And we potentially did it in the laboratory. Right now it is available in YouTube, but here I am not showing that YouTube, but it is available. This is just an example. You can see this is a water diesel mixer. At that time we didn't have crude oil. Right now we have. And we can even solidify almost 30-40 milliliters of diesel in presence of water. And you can invert this vessel without a core, because this diesel is gelled and it is solid enough to keep the water there, about 30 milliliters of water. And that's why we call them, this phase selective gelation. And this phase selective gelation is happened because the simple surfactant molecules self-organize, they self-assemble together to create a solid structure with this small fibrous structure. Under the microscope, you can see this one. And this, in this particular case, you can see this is crude oil from Gulf of Mexico. This is a crude oil. It is black, is the crude oil. Initially, it is liquid. We are adding our molecules in presence of this water, and this crude oil is selectively solidifying, and we can separate them. And these molecules are safe. It's coming from sugars and fatty acids. Now I am switched gear. I'm talking about some of the interesting new curding molecules we developed in our laboratory. And I am sure that all of you are experts more than me. Curding is historically pulling together animals using a tool. Now we are trying to utilize a chemical to pull together oil, and this, the definition for chemical curding is oil collecting agents. And in our case, all these oil collecting agents are having a hydrophilic and a hydrophobic tail, and we are trying to utilize this molecule to reduce the surface tension of water around the oil. And suppose if you have an oil spill, especially if it is thin oil spill, whether it is Arctic Ocean or it is warm ocean, and if you can apply a herder, this is the first example I showed you, This is an ocean. Oil is spilled. Oil is spreading because of the high surface tension of the water across the periphery. And in here, in the right side and left side, if you are applying the herder, the simple surfactant molecules, which can reduce the surface tension of water from 70 to dynes per millinewton per meter, you are somewhere around 30 millinewton, then this oil cannot move into forward direction. It has to retract it. And when it is retracting, it is fattening the oil. And this is a pictorial representation. Oil spill is there. We are applying these burning molecules in a solution, and it is reducing the surface tension. Oil cannot move forward. It has to come back and retract. Again, it's a schematic representation. Oil spill, applying surfactants, and it is reduced, thickening the oil. Right now, we are applying the fire, but we never do the firing in the laboratory. This is mostly fundamental science, trying to understand the science and chemistry to develop a much more biodegradable and safer. And right now you can see here it is much more pictorial representation. This is the surface tension of the sea, water, and air. This is the surface tension of the water, oil. This is the surface tension created by oil and air. And then this surface tension, total surface tension is less than 0, then the oil cannot move forward. And these are the, you may never saw the chemistry of the OP40 and thick slick. This is the OP40. This is maybe the most, earliest version, but most efficient one. But unfortunately, they happen to be silicon-based polymers. They never may be going to degrade in the water. But the thick slick is much more of a kind of a sugar-based system. And we are developing something even interesting stuff. And the reason is starting from David, James, Tim, and everybody talk about the importance of this chemical carbon is speed, low application rate, and ease of use. And therefore, and the chemistry is, or the science is here, again, the surface tension of water should be less than 0, the total surface tension of the oil and water. And must be environmentally benign. That would be wonderful. And we are trying to develop a herding molecule. In this case, the left part, the tail part, is coming from phytol. Phytol is a byproduct of chlorophyll, and chlorophyll is in the ocean biota already. And then they degrade, they make this phytol. We try to utilize the same phytol molecules, connect with a hydrophilic part. Now it is a surfactant. And this surfactant, especially, this is coming from imidazole. Imidazole, you could find even in some of the drugs. And imidazole, this, we believe it is non-toxic. We did only very preliminary tests. Interestingly, these molecules has the capability to reduce the surface tension of water to 70 to somewhere around 28 to 30s. And this can herd the oil. Now we are not doing the experiment in the ocean, but we are trying to mimic the ocean in the laboratory, utilizing artificial salt water. Now we are creating a thin oil slick here in the artificial sea, and this is Gulf of Mexico oil, crude oil. And right now we are getting a thin slick. We are just applying a millimolar solution, a 1 or 2 drops of millimolar solution in this side and this corner, only 2 drops. We are applying very dilute solution of the molecule. What we synthesized in the laboratory, you can see what is exactly going to happen. Just accelerating a little bit. Yeah, now you can see how fast this is happening. And this is exactly what Tim was mentioning about that. If you have oil and water, vegetable oil or any kind of oil in a dish, if you are applying soap, it will retract. but here also it is retracting, but we keep this one for hours, it will never come back. If it is a regular soap, it will come back because the soap will start dissolving the oil and surface tension will come back to the normal. And now we are developing some couple other systems, and this is mostly we believe could be potentially useful for warm ocean, and because there is waves are there, and here this is wavy ocean, and in this case, because the harding monolayers tend to disintegrate in sea with waves, can binding the monolayer to polysaccharides in the sea surface microlayer improve the stability and increase the dilatational viscosity? Because we need a stable monolayer in presence of a surface wave action. If it is a calm ocean, it is much more easier, and therefore we are interested to understand what exactly, how we can create a biodegradable, interesting molecule here. And again, we are talking about, and I will, especially this one, if we can utilize the ocean biota, the micro layer of the ocean, and which contains several molecules which are coming from plant chloroplast, because there is all kind of algae is there, green algae is there, the chlorophyll is there, and all these green ocean, they may contain all these kind of similar kind of molecules, and I will show you in the picture here. This is a pictorial one because there are polysaccharides, maybe from seaweeds, and it's mostly carrageenan, and there are some other molecules, and different kind of molecules are in the ocean microlayer or sea surface microlayer. And we can utilize the chemistry of this sea surface microlayer to dampen this or minimize the wave action by engineering molecules and designing molecules which are already in the systems in the chloroplast. One example is this, again, phytanic acid. Phytanic acid is a degradation product of chlorophyll, and chlorophyll contains phytanic acid. Palmitic acid is an example, and we are trying to understand how this kind of mixture of this ocean surface microlayer can be keep the bubbles, and there is kind of a viscosity of that bubbles. This is a typical example. We are utilizing two tools, especially Langmuir project. I am not going to talk anything about that one. That is one of the tools we use in the laboratory. Another one is we are using Pendant drop experiment to understand the viscosity and the ability to keep the waves. And this is exactly what is happening in chemistry-wise, because this ocean microlayer contains seaweeds. There are several kinds of seaweed. This is one example of the red seaweed, which containing lambda carrageenan. This is having galactose kind of sugars, and there are molecules similar kind of in the bilayer membrane in the chloroplast. These kind of molecules are there. We call them NGDG. And this molecule, they are interacting each other. They make very beautiful assembled structure. They can hold each other. If this is polymeric system, it is high molecular weight polymer, seaweed containing molecule. And this is the molecule which is in the bilayer membrane in the chloroplast. If we can utilize those kind of surfactants for herding application, we could be able to have a naturally occurring surfactant for herding. That is exactly what I want to show you. And here we are using small pan scale experiments again in the laboratory. This is a pan, the oil will be in the middle, and this water is there. I will show you here. This is a simple example. Here we are applying different way. It is easy for us, that's why. Now we have a ring here. Inside it is crude oil. Now we are applying this surfactant, the herding agents here, what it is developed from or identified from the surface microlayer itself. And now you can see what is happening. This is in crude oil, pure water. It is you can see the oil is going everywhere. There is no surfactant, no herding agent. Now we are using this MGDG, that symbol molecule what I showed you. And if you are playing this one, you can see, the oil is not moving because that molecule which we use can keep that oil as such. It is a kind of a reverse herding we are trying to do. And the next one, you see. And the next one, we are using a surface pressure. That means we are using small amount of herding agent here. And then the oil is, you can see here, the oil is moving. Now we do the same experiment with a different surface pressure. That means we are applying more amount of herding agents. And here we are going to use surface pressure is 41. Now we are playing this with this video. You can see it is shrinking. And this shows that this MGDG, the simplest molecule we identified from ocean surface microlayer itself, can act as a sugar-based simple molecule, which can act as a very good efficient surface collecting agent. And this is a pictorial representation. If there is an oil spill, and it can hurt these same biodegradable safe molecules. If the fire is coming, it is burning off, and we can clean. And now we are interested to work with the same kind of surfactants or herding agents in small span in the laboratory, our own laboratories. And eventually we are currently we initiated the collaboration between Corel at the Coral Research and Engineering Laboratory. I believe everybody, including David, and everybody collaborated with them. We are already started collaborating to work with them. We are interested to work with them right now. This is the molecule I was showing, MGDG. This is the byproduct. It's available in the ocean biota. And this is pan experiments successfully showed that they can be a very good efficient herding agents. Again, this is much more of a sustainable molecular design. We can create molecules and materials designed in such a fashion that they are biodegradable and safe. And finally, thank you for your attention. I'm happy to answer the questions.
Okay, great.